So why study enzyme kinetics? Right? So it gives you a quantitative description of biocatalysis, which will help you, it can also help you determine the order of binding substrates. So this example that we showed you with Michaelis-Menten is just like a single substrate binding catalysis. A lot of biological enzymes will bind more than one substrate. And you'll see examining the kinetics will help you determine the mechanism from that binding. You can elucidate acid-base catalysis, understand catalytic mechanisms, and find effective inhibitors, and understand regulation of activity. So here's an experiment that they show you an example of how to do a kinetic measurement. First, you're going to mix the enzyme and substrate, then record the rate of substrate disappearance or the rate of product formation as a function of time, which basically is the velocity of attack. Plot the initial velocity versus substrate concentration, and then change substrate concentration and repeat. So you generate a series of graphs where you have a set amount of enzyme and a set amount of concentration. All the enzyme concentrations are the same here. You just alter the substrate concentration, and you get different initial velocities, and you can plot those over time. And this is an example of a disappearance of your substrate over time. So you can plot the slope as the initial velocity, which is what we're plotting there, right? Well, that's product concentration, but it's what we're plotting here. Okay? So the ideal rate is what we just saw here, right? Your Michaelis-Menten, where velocity is going to be equal to Vmax times substrate concentration divided by the Km plus the substrate concentration. What does the Km represent? It's a good analogous for Km. Basically a dissociation constant, except this is an enzyme, so it catalyzes some reaction. So if you have a really large Km, do you think you have really good binding to substrates?
No.
No, right. It's poor binding to substrates. The smaller the Km, the better the binding to the substrates. You will see deviations in Michaelis-Menten kinetics due to limitations of measurements. Substrate inhibition, substrate prep contains inhibitors, or the enzyme prep contains inhibitors. So in a minute we'll be learning about different enzyme inhibition. Yes? Yeah, these pretty much are all non-covalent binding interactions. I mean, you can have covalent catalysis, and you will.
Yeah, that was, I guess, my question.
Yeah, but the initial binding event is non-covalent interactions.
And we shouldn't really be able to tell that by the numbers.
Right. When we actually have the catalysis, you might have covalent interactions, but those are delayed. And you'll see in a little bit when we have the different types of mechanisms for multi-substrate reactions, you can look at those too. So the effect of substrate concentration. So we see that as you add substrate concentration, you'll generate a rapid increase, a logarithmic increase in the velocity, right? But what happens to that as you increase substrate concentration? Basically, it'll level out and not increase anymore. And why is that? What are we doing over here?
Your max. That's your max.
Yeah, we're approaching Vmax, but so basically when you're over here, all of the enzyme is saturated with substrate. So your system is saturated. So as soon as that product can get off of the enzyme, there's another substrate molecule there to diffuse right on. The only thing preventing it from actually being at Vmax is basically diffusion of the product off and the diffusion of the substrate going back on. That's why you can never reach Vmax, because that diffusion takes some time, right? It's not like instant. So, and half Vmax, the concentration at which you're half Vmax is actually your Km. You want to see how you can find that? So, where are we? So basically we have VP, velocity product formation, equals Vmax times S divided by Km plus S. So let's see, what is the concentration at which we would have half Vmax? So we can put we'll solve for the substrate concentration here. So first we'll cross multiply. We have we'll divide by 1/2 Vmax. So those Vmaxes disappear then, right? We have 2 times the substrate concentration equals the Km plus the substrate concentration. So if we subtract 1 substrate from either side, this will ultimately equal Km equals the substrate concentration. So if it's pure Michaelis-Menten kinetics, half Vmax is always going to be equal to the Km. The concentration of substrate equals Km at that point. Does that make sense? So, yeah.
Is it kind of analogous to that Henderson-Haskell jump?
That's a jump.
Like some of that.
Yeah. So, I don't know if you can see that. So determination of kinetic parameters. So parameters, nonlinear Michaelis-Menten plot should be used to calculate parameters Km and Vmax. So you want to use a nonlinear. Typically what you do though is you linearize double reciprocal plot is good for analysis of 2 substrate data or inhibition. So I just showed you how you could calculate the Km is just, first you calculate the Vmax, divide that by 2, and see what substrate concentration is. That gives you the Km. But commonly what we'll see are these double reciprocal Lineweaver-Burk plots. So Lineweaver-Burk plot is the double reciprocal plot. Let me show you how we make that. So basically, you have, where are we? Again, the Michaelis-Menten equation. Okay, but what we do is we make it a reciprocal plot. So we, instead of doing like this, we do 1 over velocity equals Km divided by Vmax times 1 over the substrate concentration plus the substrate concentration divided by Vmax times the substrate concentration. Do you see how this is a linear plot now? So this will be your y, mx plus b. These substrate concentrations can go out and you just end up with 1 over Vmax. So where y equals 1 over the velocity. X equals 1 over the substrate concentration. The slope is going to be Km divided by Vmax. And your y-intercept is 1 over the Vmax. So it gives you a nice linear equation. And you can manipulate this by adding inhibitors or changing the substrate concentration to determine the mechanism of substrate binding if you have multiple substrates, or you can determine the mechanism at which the inhibitor works. Because we'll introduce inhibitors here in a second. So derivation of enzyme kinetic equations, start with the model mechanisms, identify the constraints and assumptions, carry out the algebra, or graph the theory for a complex reaction. And the simplest model mechanism is what I just showed you, right? One reactant, one product, and no inhibitors. And we just went through this a little bit before, so I'll skip over this. But the final form of a single substrate is this, right? They have the K-cat, which is basically your K2, and that's all dependent on the mechanism. You can't see it back there, but it's K2. So K-cat times the enzyme total times substrate divided by Km plus substrate. K-cat's the turnover number, which is how many substrate molecules one enzyme molecule can convert per second. So, and Km is Michaelis-Menten constant. It's an approximate measure of the substrate's affinity for an enzyme, basically the dissociating enzyme. Microscopic meaning of Km and K-cat depends on the details of the mechanism, basically. Enzyme efficiency is measured by dividing the K-cat by the Km. So you can gain efficiency by having a high velocity or affinity for the substrate. So you can look at catalase versus acetylcholinesterase here in your figure. If you look at acetylcholinesterase, it has a K-cat of 1.4 times 10 to the 4. What's that? 14,000 molecules it'll convert per second. And the Km is 9 times 10 to the minus 5 molar. So that's relatively small, so it has a relatively high affinity for acetylcholine, breaks it down into choline and acetate, basically. And then, so the efficiency is 1.6 times 10 to the 8th, so fairly high. If you look at catalase H2O2, basically that's 4 times 10 to the 7 K-cat. What is that? 40 million molecules per second that it'll convert. Whereas look at its Km, 1.1 molar. So is that a high affinity or low affinity binding? Low. So remember, big Kms equal low binding affinity. Really small Kms like this, really high binding affinity. So this has a really large K-cat and a really low binding, but it still has relatively almost as efficient as cholinesterase. So it shows you how two different. This one has a high binding affinity, 10 to the minus 5, and a somewhat okay K-cat, whereas this is really fast, but really low binding affinity. Thing is, it's so fast, it immediately converts peroxide to water, basically, and oxygen. So it breaks it down really fast. So kinetic mechanisms of a two-substrate reaction. Most of the enzymes we're going to see are two-substrate reactions. And the order of binding substrates and release of products is how the mechanisms are determined. You can have 2 or more reactants involved in the enzyme kinetics, allows to distinguish between different kinetic mechanisms. You have a sequential mechanism versus a ping-pong mechanism. These are the figures that show you the different mechanisms. You have a random order where it doesn't matter. Your enzyme can bind to the substrate 2 or substrate 1 first, and then it'll bind to the other substrate to form an ES-S complex that yields your E and your product 1 and product 2. Or you can have an order where the enzyme specifically has to bind to the first substrate in the correct order. Then it'll, form an ES complex, then it'll be able to bind, have affinity for substrate 2, which will then initially yield your products. Or you have your ping pong mechanism, where the enzyme reaction in which no ternary complex is formed. The enzyme binds to substrate 1, forms product 1 and a modified enzyme, which then has affinity. It'll release product 1, and the modified enzyme will bind to substrate 2, producing product 2. So it's like ping pong, where it's substrate 1 on, product 1 off, and then substrate 2 on, product 2 off. And then it returns the enzyme back to its original state. So different mechanisms. And these can be determined with this Lineweaver-Burk plot. So cannot easily distinguish random from ordered, but random mechanisms in equilibrium will give an intersection point at the Y value, and the lines will intersect. As you increase substrate 2, substrate 1 is constant, you see it alters the slope of the line. So if it alters the slope, what's changing?
S2.
So slope represents Km and Vmax. So what's changed, and the y-intercept is 1 over Vmax, right? So Vmax is changing, and so is Km. Negative, the x-intercept is going to be your negative Km value. So you can see that they're both changing in this case. Ping pong kinetic mechanisms, the lines are always parallel. So as you increase S2, substrate 2, you keep, as you increase the concentration of substrate 2, you see that the Km over Vmax is changing, whereas Vmax is changing also. The slope's always the same. So enzyme inhibition. Inhibitors are compounds that decrease the enzyme's activity. And there's a number of different classes. We have irreversible inhibitors or inactivators. They react with the enzyme itself. So they form a covalent bond typically. A lot of those, you've heard of suicide inhibition. One inhibitor molecule can permanently shut off one enzyme molecule. And they're powerful toxins, can also be used as drugs. So that's where the inhibitor itself reacts. It's involved typically they're designed after the reaction mechanism, and they'll form a covalent bond with the enzyme, killing it for good. Reversible inhibitors will bind to and can dissociate from the enzyme. They're often structural analogs of substrates or products, and they're often used as drugs to slow down a specific enzyme. So a reversible inhibitor can bind to the free enzyme and prevent the binding of the substrate to the enzyme substrate complex and prevent the reaction. So different types there. First we'll look at is competitive inhibition. So you can think of it competes with the substrate for the binding. So typically it will bind to the active site. That inhibitor and the substrate are going to compete for binding for that active site. So it does not affect the catalysis at all. It's just you're flooding it with something that won't react in the enzyme, but still wants to bind in that site. No change in Vmax, but you have an apparent increase in Km. It's not a real increase in Km, but it's having the inhibitor present at a set concentration will make the apparent Km seem larger. So, because you have something there competing for that active site. Lineweaver-Burk plot, the lines are going to intersect at the y-axis. So, it's pouring. So this is your Lineweaver-Burk. So this is without inhibitor, and this would be with, right? The Vmax stays the same, so that's why it intersects there. But what's increasing is your Km is getting bigger, the Vmax stays the same, so you you see an increase in the plot like that. Glad I rolled up my windows when I did.
So is sequential kinetic mechanism always going to be competitive and ping pong always uncompetitive?
So those mechanisms, they're not competitive. We're talking about inhibitors now, basically. Wait,
Because it says for the Lineweaver-Burk, it says the lines intersect at the y-axis. That's why I was trying to connect those two.
Yeah, so if you're looking for the mechanism, so for the mechanism, the random mechanism in equilibrium will give an intersection point at the y-axis. So it'll look similar to this, but we're not adding an inhibitor. For this plot, we're adding substrate concentration, one of the others. So basically, yeah, altering the K. And this is basically competitive inhibition. You have an inhibitor in the mix, enzyme, free enzyme, and substrate, which can form its enzyme-substrate complex and then go on to make the product. Or the enzyme can bind the inhibitor and form an enzyme-inhibitor complex. So when you add inhibitor to the mix, you'll shift this equilibrium this way because you're taking enzyme out of the system and forming this enzyme-inhibitor complex. But as you increase substrate concentration, you can outcompete for this binding site for the inhibitor. So eventually your Vmax will stay the same. If you just flood it with substrate, eventually you'll end up with the same Vmax. Flood it with enzyme, you'll shift that substrate, you'll shift the equilibrium to the product formation. Eventually you'll outcompete this inhibitor. Does that make sense? Whereas competitive inhibition, wait, this is competitive inhibition. That's fine. This is the same plot I just drew you. Vmax always stays the same, so 1 over Vmax will be the same, or your y-intercept. As you increase the concentration inhibitor, you increase the apparent Km. They put this little alpha here because it includes a fudge factor for the concentration of inhibitor that's been added. Does that make sense? Uncompetitive inhibition only binds to the ES complex, does not affect substrate binding, and inhibits catalytic function. So you have a decrease in Vmax, an apparent decrease in Km, no change in the Km divided by Vmax or your slope. Lineweaver-Burk lines are going to be parallel. And in this model, we have our enzyme plus substrate forms an enzyme-substrate complex. This is the only thing that can bind to the inhibitor in this model to form this ESI where you'll inhibit the reaction from taking place, or you can end up with your product formation. So in this case, as you increase inhibitor concentrations, we do shift your Vmax and your Km values. So the Km gets large, or
Km gets shifted.
huh?
The Km gets shifted.
Yeah, the Km gets shifted and the Vmax gets shifted, but to equal degrees, so the slope is always constant. So you have parallel lines for your uncompetitive inhibition. And this isn't something you really see in nature. So typically, if you're going to have something that's going to bind to an enzyme substrate complex, it will also bind to the free enzyme. And this is more of what we'll see as mixed inhibition. Okay? So the enzyme plus the substrate plus the inhibitor, you can form a free enzyme inhibitor complex, which can then go on and bind the substrate. And if these two rates are the same, like if it has equal affinity for the free enzyme plus the enzyme substrate complex, it's called pure non-competitive inhibition, but most of the time it's going to be mixed inhibition. And what's happening here, this is basically when you bind the enzyme or the enzyme substrate complex with the inhibitor, you take these out. So what you see is a decrease in Vmax. And you'll also see an apparent change in Km if those are not equal. So the Lineweaver-Burk lines intersect from the y-axis. Non-competitive inhibitors are mixed inhibitors such that there is no change in Km. So it looks something like this. And then your Lineweaver-Burk plot will look like this. So you increase the inhibitor, you're going to decrease the Vmax, and at the same time, for mixed inhibition, you're going to alter the Km also. So which type of inhibitor pretty much binds at the active site? Competitive inhibitor. So mixed inhibitors or non-competitive inhibitors typically bind somewhere away from the active site, and they're just going to alter the enzyme's ability to perform its catalysis, basically. Does that make sense? So it changes the shape of the availability of the active site to perform the catalysis. So enzyme regulation. So enzyme regulation of enzyme activity can result from non-covalent modifications or covalent modifications, reversible or irreversible. I said those backwards. So non-covalent modifications, one example of non-covalent modifications is allosteric regulation. Right? So in allosteric regulation, basically you have two states of the enzyme. Right? The high activity R state, which stands for what? Relaxed. Or a low activity tense state. And typically you have non-Michaelis-Menten kinetics because there's basically two states of the protein, and you'll see a sigmoidal type curve which indicates what? So typically these enzymes are multi-subunit enzymes. You get binding of the substrate by one subunit is going to increase the affinity for the next subunit. What's that called? Come on, you guys can be positive about this. Work together a little bit. Positive cooperativity, right? So when you see sigmoidal shape, it's positive cooperativity. As you increase the concentration of substrate, the activity is going up, so your affinity for that substrate is increasing like that. So you end up with what's known as, it's kind of like mixed Michaelis-Menten kinetics between the two states, basically. Some reversible covalent modifications. So these are examples of modifications to enzymes. Enzymes have sites, they're proteins. They can be phosphorylated. Do you know what enzymes phosphorylate proteins? Kinases. And the enzymes that remove phosphate groups or dephosphorylate proteins are called phosphatases. So adenylation, can you see this? So this is basically, you add an AMP to an enzyme. That's another covalent modification that can occur. Acetylation is where you add an acetate group you have an acetate ester bond. Myristerylation, it's where you add myristeryl-CoA. Ubiquitination, didn't we talk about ubiquitination? If you remember, those are like little peptides that are added to a protein. What happens when a protein gets ubiquitinated? It's a signal for that protein to be destroyed, right? So it's part of that proteostasis thing. Proteins are tagged for destruction with ubiquitin, and then it gets targeted for destruction. So activated ubiquitin there. ADP ribosylation. So here we have an enzyme, and you have ADP added to it. And then methylation, of course, you can add methyl groups to enzymes. Another modification that regulates enzymes are Zymogens. So Zymogens are activated by irreversible covalent modification. And some examples of these are blood clotting enzymes, also our digestive enzymes. So chromotripsin and trypsinogen are both inactive in their pro form. So you have the whole protein, it's produced in your pancreas, right? And it's going to be secreted into your small intestines to digest your dietary protein. So you don't want an active protease then in your system, digesting your own proteins. You want it inactive when it's first produced, and it's first produced as a full-length protein, or your protrypsin, or chromotripsinogen. Same thing with trypsinogen. They put the O-gen there to indicate that it's the pro form of the protein. So trypsin will cleave chromotripsinogen, and you'll end up with a peptide fragment on the N-terminal side along with pi chromotripsin. That gets cleaved again by pi, but auto-cleaves, and then you end up with alpha chromotripsin, this is active, and this is an active form of chromotripsin. So chromotripsin is your protease that digests peptides following aromatic amino acids, right? Do you remember the mechanism chromotripsin has? What class of enzyme is this?
Autolysis.
Huh? Well, yeah, autolysis, but what's the mechanism at the active site? We discussed it, I think, last time a little. It's acid-base catalysis along with covalent catalysis. It's one of your serine, but actually both of these are serine proteases. Can anybody tell me what the catalytic triad is? So those are three amino acids found at the active site of all serine proteases, right? You have a serine, a histidine, and an aspartic acid, right? Aspartic acid draws a proton away from the histidine, which then draws that proton away from the alcohol group on the serine, allowing the oxygen on that alcohol group to attack the carbonyl on a peptide bond, right? That forms a tetrahedral intermediate, which collapses. You release one, the new N-terminal peptide that was formed, and then water will come in and cleave the rest of that, the new C-terminal form of the peptide. So the blood coagulation cascade uses irreversible covalent modifications also. And you guys are going to learn this cascade when you take hematology, I'm pretty sure, right? Do you guys know it at all? You probably had to learn it for anatomy and physiology, did you?
We didn't have to learn, not for Chef.
No?
There's a little bit of it in the book, where it just goes through the intrinsic pathway and versus the extrinsic pathway.
So a lot of these, if you look, so prothrombin gets cleaved to make thrombin. So this is a zymogen of thrombin. And then thrombin will cleave, what do we have here?
8.
Factor 8, that's 13, right? 13 to 13A, which then cross-links fibrin. Thrombin also cleaves fibrinogen into fibrin monomers, which will then form a polymer and then gets cross-linked by factor 13A to form your clots, basically. But a lot of these are modified also irreversibly. Some enzymes use multiple types of regulation. Actually, most enzymes you'll see in the pathways when we get to the metabolism are going to use multiple forms of regulation. So this is an example of phosphorylase kinase. And to the active phosphorylase kinase basically is modified with a phosphate group. So phosphorylase kinase is going to phosphorylase phosphorylase B to form phosphorylase A. Gets dephosphorylated by phosphorylase phosphatase 1. Okay, and this shows you all the different regulation mechanisms. So phosphorylase A is activated by AMP, deactivated by ATP, or inhibited by ATP, inhibited by glucose 6-phosphate rather. Whereas phosphorylase B is inhibited by glucose 6-phosphate, ATP, and AMP. And this is set up with insulin regulation, right? So energy regulation. So insulin, can somebody tell me what insulin does in the body? So it does more than like regulate sugar, basically regulates energy. So insulin is pro-energy storage. So energy is going to be for storing energy. Versus glucagon is pro-energy usage, basically. So it is for regulating blood sugar, insulin is. If you have too much blood sugar, it's time to store that sugar. If you have too little blood sugar, you should produce glucagon, which is going to be a signal to release more sugar out of the blood, or use the sugar that you have. So you're going to release sugar from glycogen, and you'll also start to produce sugar and release it into the blood. So glucagon will increase cyclic AMP, which will then activate protein kinase A, which phosphorylates phosphorylase kinase, which is, and when we're saying weird cramp. So, which will then cause this phosphorylase is an enzyme. It breaks down glycogen basically in the muscle. So it splits those glycogens apart, and it's a phosphorylase because it uses the phosphate in the glycogen uses phosphate to split it apart. So you end up with glucose 6-phosphate right off the bat, which happens to be one of the products here. So phosphorylase kinase is the enzyme that activates phosphorylase. So phosphorylase A then gets activated by phosphorylase kinase, which is activated by protein kinase A. So via an extracellular single glucagon. And then phosphorylase A is regulated not only by AMP and ATP, and these are allosteric regulators of phosphorylase A, because AMP and ATP does not work on this, but it's a way for the enzyme to sense your energy state in the cell. If you have a high energetic state where you're not going to need to burn sugar, are you going to have a lot of ATP or AMP around? Yeah, high energetic state of the cell, you're going to have ATP. So ATP will want to inhibit phosphorylase A, so you're not breaking down your glycogen when you don't need it because you have lots of ATP around. Does that make sense? And the opposite is true if you have lots of AMP around, means you're low in ATP. So that will activate, this is an allosteric activator of phosphorylase A, which will cause an increase in the production of glucose 6-phosphate from glycogen. And then glucose 6-phosphate is the product of the reaction that catalyzes. It'll actually inhibit its own production. This is active for a while, and you have a buildup of glucose 6-phosphate, it'll inhibit, so you're not producing it. So this is an example of multiple types of regulation on one enzyme. And that's the end of chapter 6. Now we can go to sugars. So some key topics about carbohydrates are we'll look at the structures and the names of monosaccharides, open chain and ring forms, structures and properties of disaccharides, biological functions of polysaccharides, and biological functions of glycoconjugates. So carbohydrates are named basically because they all have the same formula. They're basically the hydrates of carbon. So for every molecule of carbon, you're going to have one molecule of water in a carbohydrate. No matter what kind of carbohydrate it is, you'll always follow this formula. So, and then they're produced, of course, from carbon dioxide and water via photosynthesis in plants. Range from small, like glyceraldehyde, to as large as amylopectin, with a molecular weight of 200 million grams per mole. Fulfill a variety of functions, include energy source and energy storage. That's what we normally think of when we think of carbohydrates. They have structural component, the cell walls and exoskeletons. So cell walls are made up of cellulose, which is a polysaccharide of glucose. Exoskeletons are made up of chitin, which is it's like a glucose with an amine, a methylamine group on it. Informational molecules and cell-to-cell signaling. So our blood typing is has sugar involved. Different antigens there on our blood cells. Can be covalently linked with proteins to form glycoproteins and proteoglycans. So a lot of our extracellular space is full of proteoglycans that interact up there. So there's some main types of carbohydrates. You have aldoses and ketoses. So aldoses contain an aldehyde. Glyceraldehyde is an aldotriose, or 1, 2, 3 carbons, so it's a triose. Dihydroxyacetone is a ketriose. It's basically, these will have the same molecular formula, right? But one's an aldose and one's a ketose. And you guys remember the differences between aldehydes and ketones, right? So, pretty simple. So, glucose is a aldohexose, basically, because it's an aldose. Fructose is a ketohexose, because it has 6 carbons long. You guys remember enantiomers? Basically, superimposable stereoisomers. So in sugars that contain many chiral centers, which most sugars have many, the only one that is the most distant from the carbonyl carbon is designated as the D or L. And the D or L isomers of a sugar are enantiomers, so L and D glucose have the same water solubility. They're enantiomers of each other. But most hexoses in living organisms are D stereoisomers. So we're going to be working with D sugars. And I think there's a table in your book with all the different D monosaccharides. So some simple sugars occur in the L form, such as L-arabinose, but the majority we're going to see are D. This is the ball and stick model showing you stereoisomers. If you guys remember that from, I think, organic is when they introduced that. And chiral compounds can be drawn using perspective formulas. However, chiral carbohydrates are usually represented by Fischer projections, where the horizontal bonds are pointing towards you and the vertical bonds are projecting away. And this shows you your Fischer projections. And this is typically how you see it. This is D-glyceraldehyde, where there's your stereocenter. The alcohol's on the right versus the L-glyceraldehyde, the alcohol's on the left there. And these are your perspective formulas showing you the same thing. Diastereomers are stereoisomers that are not mirror images, and we'll see that a lot of them, like aldoses, D-erythrose, D-threose, a lot of them are diastereomers. So if you notice, they have the same orientation here. They're both D. This is what designates the D, is the orientation around this carbon. Whereas this, they'll just get their other names for these stereocenters. So you'll see D-glyceraldehyde only has, this is the only center that has optical activity, whereas erythros and threose have 2 carbons. So epimers are 2 sugars that differ only in the configuration around 1 carbon atom, such as like mannose and glucose or galactose. So D-glucose, it only differs around carbon 4, with D-mannose around carbon 2. So epimers, they only differ around one of the stereo centers. Some structures to know would be ribose, it's a standard 5-carbon sugar. Glucose is their standard 6-carbon sugar. Galactose is an epimer of glucose. Mannose is an epimer of glucose. And fructose is the ketose form of glucose. So fructose is a ketone. These are your D-aldoses, 5 carbons. And if you notice, they're all D. If these were switched around this first carbon, they would be L, but they're all D because of this carbon. And then they get their different names based on where they differ in orientation around these other carbons. So here's D-glucose, D-mannose. Obviously only differs around this one. D-galactose, it's only different around this one. Then your D-ketoses, dihydroxyacetone. So this is the only, no optical center on this molecule, right? Whereas D-erythrose, erythrulose, I can't say these. And then there's fructose. So we'll see, we convert glucose into fructose before glycolysis, or during glycolysis, right? So we'll see how that is done in the future. Then hemiacetals and hemiketals, so the aldehyde and ketone carbons are electrophilic. So these are the reactive parts on the sugar, right? Alcohol oxygen atom is a nucleophilic, and when aldehydes are attacked by alcohols, you can form hemiacetals. When ketones are attacked by alcohols, we form hemiketals, right? So if you have an alcohol, will act as a nucleophile, attack the ketone, and you end up with an acetal. Same thing with the, what did I say, ketone? Mm-hmm. When the alcohol reacts with the aldehyde, you form an acetal, or a ketone reacts with the alcohol, you form a ketal. So, and we'll see how this is important for the cyclization of monosaccharides. So pentoses and hexoses readily go intramolecular cyclization. The former carbonyl carbon becomes a new chiral center called the anomeric carbon. So the anomeric carbon in a sugar is always where that aldehyde or ketone is. So the former carbonyl oxygen, and it'll become a hydroxyl group. The position of this group determines if the anomer is alpha or beta. The hydroxyl group is on the opposite side, or trans. The ring, the CH-OH moiety is configured as alpha. If the hydroxyl group's on the same side, or a cis, where both hydroxides are pointing up, the ring, its moiety, its configuration is beta. And I think this next figure shows you pretty clearly. So you have D-glucose and the aldehyde, the free aldehyde form. The alcohol here on the number 5 carbon will act as a nucleophile, and it'll be able to form two different structures: beta D-glucopyranose. Pyranose is just a 6-ring form of a sugar, a pyran ring, or alpha D-glucopyranose, where if you see this carbon's projected up, the alcohol's projected down, or like trans. So trans is alpha. Beta, they both project this, the alcohol's up, and so is this carbon. So that's how you can determine whether the ring form is alpha or beta. Basically, trans is alpha, cis is beta. So try to remember that. And then the anomeric carbon is always this carbon with the aldehyde, or the number 1 carbon. Or if it's a ketose, it'll be the number 2 carbon. So pyranoses and furanoses. So 6-membered oxygen-containing rings are called pyranoses. 5-membered oxygen-containing rings are called furanoses. Anomeric carbon is usually drawn on the right side. And just to show you something, anytime you have glucose in solution, it transitions back and forth between these structures regularly. So it can convert back between the aldehyde form of glucose. Instead of, if this alcohol group, if it wasn't this alcohol doing the attacking, but let's say this alcoholic group attacking the carbonyl, which can happen also, you can form glucofuranose rather than pyranose. It's called pyranose because this is the more common in glucose than the furanose. Basically because the furanose is going to be more restricted.
How does the other alcohol attack the carbon? It's not.
This one would act as the nucleophile instead of this one. This is full of alcohol groups.
Right.
So the equilibrium favors the 6-member ring because the 5-member ring is more constrained, right? It's whereas the 6-member ring is less constrained. But you can still form a furanose with glucose.
Okay.
So it just will happen randomly. But the thing to remember is this is an equilibrium process going back and forth between the alpha, beta, furanose, and pyranose forms. So, and there will be equilibrium constants for all of those. So, and what this is called when it does this back and forth, it's mutual rotation. If you've heard of that before. So this is glucopyranose, beta D-glucopyranose. So this is alpha D-glucopyranose, and you see that the number 6 carbon is projected up, and the number 1 alcohol or on the anomeric carbon is down, so it's alpha. Beta D-glucopyranose, and this is a purine ring, or your furan ring. And the most common structure in fructose is to have the furanose. So alpha D-furanose, so the anomeric carbon is the number 2 carbon because it's a ketose. And alpha, see this is projected up, whereas the alcohol is projected down, so it's alpha versus beta. The alcohol is projected up along with this number 6 projected up. So chainring equilibrium, I just talked about this. So the ring exists in equilibrium with the open chain forms. Aldehydes can reduce copper via Fehling's test, and aldehyde can reduce silver. Tolling's test allows detection of reducing sugars such as glucose. So it's that the free aldehyde or free ketone is what's going to make it a reducing sugar. These are some colorimetric of glucose analysis. So you have glucose oxidase will react with oxygen to produce H2O2, and gamma D-gluconolactone, and that will react with peroxidase, form water, and you oxidize this substrate to this via peroxidase, and you'll end up with a color, a bright orange color that you can measure. That's how that test works. And then these are hexoses because that these alcohols, they can be replaced, right, or derivatized. So you have glucose or glucosamine. So this is the beta D-glucosamine. Those two are up, and then you have an amine group substituted somewhere. This is muriamic acid, or you have another R group here, or N-acetylmuriamic acid, where they're acetylated. The N groups are acetylated, so it's N-acetylmuriamic acid, or N-acetyl-beta-glucosamine, and so forth. Where they can be derivatized. You have amino sugars, which are derivatized with the amino group. Deoxy sugars, they're lacking one of the alcohols. L-glucose or alpha-L-ramose or deoxy sugars. If you notice that they're lacking oxygen on their number 6 carbon. Deoxyribose and DNA is lacking a sugar also. Beta-D-glucouronate means it's been oxidized and you have a carboxylic acid. D-gluconate, oxidized again, and then D-glucogamma-lactone. So you're forming a lactone there between the two. So the glycosidic bond is when 2 sugar molecules can be joined via glycosidic bond between an anomeric carbon and one of the hydroxyl carbons. So the glycosidic bonds in acetyl between monomers is less reactive than the hemiacetal at the second monomer. Second monomer with the hemiacetal is reducing. The anomeric carbon involved in the glycosidic linkage is non-reducing. Do you understand what that means? So you form a disaccharide between the anomeric carbon, one, it reacts with an alcohol on another sugar. So that alcohol acts as, I can't breathe. So the alcohol that forms from the anomeric carbon, I think there's an image here. So basically, this is your anomeric carbon of glucose and the alcohol of beta-D-glucose. You get condensation, and you have an acetal between the two. And this is a hemiacetal still. So this second sugar molecule will be able to switch back and forth between the beta and the free anomeric form, the free aldehyde form, and then the alpha. So which is what makes this reducing, because this aldehyde here is still available for reaction. This one's tied up in the glycosidic bond. Does that make sense? So non-reducing disaccharides is when 2 sugar molecules can be joined via a glycosidic bond between the 2 anomeric carbons. So you'll see that with fructose. So the product has 2 acetyl groups with no hemiacetals. There's no reducing ends because you can't, because the second sugar, this is the anomeric carbon here. If it was bound to this alcohol, it wouldn't be able to do that mutual rotation between the free aldehyde, the alpha and beta anomers, right? So there's no non-reducing ends, so it's a non-reducing sugar. Trehalose is a constituent of hemolymph of insects, provides protection from drying. It's an example in resurrection plant. And then fructose, or sucrose is a non-reducing sugar also. So lactose, and I didn't tell you how these are named either. So you have beta lactopyranose 1 to 4 beta glucopyranose. So this one has a beta 1 to 4 bond. Do you see that? This one has an alpha 1 to 2 bond. And then this has an alpha 1 to 1 bond here. So polysaccharides are natural carbohydrates. They're found as polymers. And polysaccharides can be homopolysaccharides, which means they all contain the same sugar over and over and over again. Heteropolysaccharides contain multiple different sugars. They can be linear or branched. And then polysaccharides do not have a defined molecular weight because it's a polymer. They're going to be different molecular weights. This is in contrast to proteins because unlike proteins, no template is used to make a polysaccharide. You just keep adding sugars to make a starch or a glycogen or so forth. So this is the difference, branched, unbranched, and branched. So unbranched, they're going to have the same type of bonds all the way through in a linear fashion. So these could all be alpha 1 to 4 polyglucose. That would be just normal amylose. You could have alpha 1 to 4 here, but then here's a 1 to 6 bond, where it is a branch site that's common in amylopectin or glycogen. Or if you have 2 different types of sugars, you can have a heteropolysaccharide, where you have different types of sugars in a polysaccharide, and they can be branched also. So glycogen is a branched homopolysaccharide of glucose. Glucose monomers form alpha-1 to form linked chains, and branch points occur at alpha-1 to 6 linkers every 8 to 12 residues. So a branch for glycogen. Molecular weight reaches several millions and functions as the main polysaccharide mammal. So can somebody tell me why would we want branched polysaccharides as an energy storage?
Well, because you probably put those branches at the weak point and you have multiple mono.
Well, don't think about the branches at a weak point, but at the end of each branch, there's 1 glucose. If you have multiple branches, there's a ton of different single glucoses that are available right at the end. So you have a bunch of, more than 1 glucose you can pull off at a time because of all the branch sites, right? And then you can harvest a lot of free sugar all at once rather than having to pick it off one by one if it was a linear polysaccharide. Starch is a mixture between 2 homopolysaccharides of glucose. Amylose is just linear alpha-1 to 4 linked glucose. And amylopectin is similar to glycogen, but it doesn't have as many branches. So amylopectin has alpha-1 to 4 main chain linkages with alpha-1 to 6 linkers every 24 to 30 residues. Molecular weight of amylopectin is up to 200 million. And this is the main polysaccharide storage in plants. So glycosidic linkages in glucose and starch, they all have alpha-1 to 4 main chain linkages, and this is amylose, whereas glycogen and amylopectin will have a branch point at alpha-1 to 6. And you can see it's alpha because you have the anomeric carbon here. This is going down, this is up, so it's trans, so it's alpha, and then it's linked to the number 6 carbon of this glucose, so alpha-1 to 6. See how that works? So mixtures of amylose and amylopectin in starch. So you can have both in starch. You have a mixture. I don't know what this figure is supposed to do. So metabolism of glycogen and starch. Glycogen and starch often form granules in cells. Granules will contain enzymes that synthesize and degrade these polymers. Glycogen and amylopectin have 1 reducing end but many non-reducing ends. So the enzymatic processes occur simultaneously in many non-reducing ends. And that's what I was just saying, how you can cleave off a number of those non-reducing ends at once. And the enzyme that does that is a phosphorylase, or glycogen phosphorylase, what we just went over a minute ago. Cellulose is a branched homopolysaccharide of glucose, and glucose monomers form beta-1 to 4 linked chains. So the only difference is that cellulose has 2 glucose, but it's a beta-1 to 4. So they're both cis, right? And what this does is it alters how that is going to form. Amylose or starch typically likes to form helices, whereas cellulose is more of a linear type structure that flips up and down, which results in additional hydrogen bonds between the chains, and you form this very tough water-insoluble structure. It's the most abundant polysaccharide in nature. Cotton is nearly pure fibrous cellulose. So this is the beta-1 to 4 linked D-glucose unit. You'll notice this is your anomeric carbon, and it's beta because this is going down. It's hard to tell what number is what. Oh, but this is 1, 2, 3, 4. This would be 1, 2, 3, 4. So it's kind of flipped in this image, so it's hard to see. So it's beta 1 to 4 in here. And then they form these linear chains and you get hydrogen bonding between them, between the chains, which makes it really tough. And you'll also get hydrogen bonds stacking on top of each other too, but they don't have a figure showing you that. So the fibrous structure and water insolubility make cellulose difficult substrate to act on. Fungi, bacteria, and protozoa secrete cellulase, which allows them to use the wood as a source for glucose. And most animals cannot use cellulose as a fuel because they lack cellulase, which hydrolyzes beta 1 to 4 linkages. However, ruminants and termites, we all know they live symbiotically with bacteria that will produce cellulase. Cellulases hold promise for biomass and biofuels, right? Chitin is the linear homopolysaccharide N-acetylglucosamine. So N-acetylglucosamine monomers form beta 1 to 4 linked chains, and they form extended fibers, which are similar to those in cellulose. But this is chitin. So it's what the shells on insects is made out of, or the shells on crustaceans also. Hard, insoluble, cannot be digested by vertebrates. Structure is tough but flexible and water insoluble. Found in the cell walls of mushrooms and exoskeletons, insects, spiders, crabs, and other arthropods. And this is the structure basically of chitin. So you have your N-acetyl glucose basically, and it's formed beta 1 to 4, right? So this is going down, and then this is going kind of up, and this is up. So it reverses up and down, but they're all together. And this shows you a picture of a bug. That shell is made of chitin. So agar and agarose. So we use agar to make our gels, right? Or agarose rather. Agar is a complex mixture of heteropolysaccharides containing modified glycogen. Agar serves as cell wall and some leads. And agarose is one component of agar. So we use purified agarose to make our gels and agar to make plates, right? Agar solutions form gels that are commonly used in the laboratory, and agarose is used to make, separate DNA by electrophoresis. This shows the structure of agarose. It's D-galactose beta 1 to 4, 3,6-anhydro L-galactose 2S alpha 1 repeating units. So it's sulfonated. And again, this is galactose beta 1 to 4. So this is projecting up from the anomeric carbons, the number 1 carbon. And then the number 6 carbons projecting up too. So that's a beta 1 to 4.
Then it's alpha 1 repeating units.
Yeah.
So the O, the 1 to 4 link is...
So this is actually a cyclic structure here, but it's projecting up, and then this is back down again. You see that? So glycosaminoglycans are linear polymers of repeating disaccharide units. One monomer is either going to be N-acetylglucosamine or N-acetylgalactosamine. They typically are negatively charged, and uronic acids, C6 oxidation occurs, sulfate esters, and extended hydrated molecule minimizes charge repulsion because of all the hydration, forms a meshwork with fibrous proteins to form our extracellular matrix. We see it in connective tissues and lubrication of joints. So when you squeeze these polymers, the water can be squeezed out, and then when you press on them, the water will come back in. So that's some of the shock we get from our joints, connective tissue. And then glycosaminoglycan, so this is hyaluronate. So it's got this beta 1 to 3 structure, and then glucose and acetyl, and then chondroitin 4 sulfate has got that beta 1 to 3 also. So, are we about out of time now?
We end at 3:40.
Oh. Why do I always think it's 3:30? Huh?
It can't be flat.
So heparin and heparin sulfate. Heparin is a linear polymer, 3 to 40 kilodaltons. Heparin sulfate is like heparin, like polysaccharide, but it's attached to proteins. The highest negative charge density biomolecule. Prevents blood clotting by activating protease inhibitor antithrombin. And it'll bind to various cells, regulating development formation of blood vessels. It can also bind viruses and bacteria. So, in fact, one of the ways you purify adeno-associated viruses, you make an affinity column out of heparin sulfate, and that'll bind up your virus, so you can purify it from, it's cell lysate that you've made the virus with. And this is the structure heparin subunit. So the interaction between glycosaminoglycan and its binding protein looks like this. And then structures and roles in some polysaccharides. So this is a figure from your book. Amylose, it's a homopolymer, meaning that it's all glucose, right? Amylopectin is the branched form of starch, and amylose is linear. It's energy storage in plants. Glycogen is homopolymer glucose again, energy storage in bacteria and animal cells. Cellulose is a homopolymer of glucose, beta 1 to 4, which alters it from a helical shape to a linear shape, making it very strong via interchain and intrachain hydrogen bonds that you get, and it's structural in plants, gives rigidity and strength to the cell walls. Chitin, again, is very large, homopolymer of beta 1, 4 N-acetyl glucose. It's very large, and it gives structure, and it also has a linear type form. So you get interchain and intrachain hydrogen bonding to form the exoskeletons. Dextrin is something we didn't talk about. It's similar to amylose. So it's a homopolymer of glucose, but rather than the main chain being alpha 1 to 4, like you see in amylose or amylopectin, the main chain is alpha 1 to 6 glucoses with branches on alpha 1 to 3. So, and anybody know where you find dextrins in your body?
Bacteria is extracellular.
Yeah, the results from bacteria. Oh, you're reading it there. But where do you find it in your body?
Well, dextrose is a sugar. You get it with.
It's not dextrose. Yeah, dextrose just stands for D-glucose.
Yeah.
But dextran is a polysaccharide of D-glucose, and you find it on your teeth. That's that plaque you have a hard time getting off your teeth. Is the bacteria that lives in your mouth will produce dextrins, and it sticks to your teeth as dextrin. So that's the structure. The plaque on your teeth is dextrin. Peptidoglycan can be hetero. Peptides can be attached. They're very large. They're in bacteria, give rigidity and strength to the cell walls. Agarose, that's what we make our gels from algae. And hyaluronic acid, glucosamine glycan. It's hetero again, and it's structural in vertebrates. So, glycoconjugates, also like glycoproteins, is a protein with a small oligosaccharide attached. Carbohydrate is going to attach via its anomeric carbon. About 500,000 proteins are glycoproteins. Carbohydrates play a role in protein-protein recognition. And so normally your glycoproteins are going to be extracellular or transmembrane proteins with the extracellular domain glycosylated. So only some bacteria glycosylate few of their proteins. Viral proteins are heavily glycosylated, and this helps them evade the immune system. So, and basically, so if you have a cell membrane, so a lot of these glycoproteins they're embedded in the membrane. And then you have extracellular domain. And it has to get out. You'll have a globular domain up here. Have it going straight. And this is the domain that has all the glycosylations on it. And that's what helps keep, it extends basically this extracellular domain out away from the cell. So you get this globular domain that can bind to its, ligand projected away from the cell, basically. Because adding all these sugars is going to add water solubility to that part of the peptide chain, basically. Does that make sense? So, two types glycosylations you can see, and you have O-linked galactose N-acyl groups, O-linked. So they're going to be O-linked to a serine or a threonine, or you can have N-linked, where you have not asparagine, the amide, N-linked, where you have sugars N-linked to that. So N-linked, it's attached to the amide of asparagine. O-linked, it's attached to a serine or a threonine, to the alcohol group. And if you see it, notice it's the anomeric carbon that's attached. So a lipid with a covalently bound oligosaccharide. So glycolipids, part of the plant and animal cell membranes. Invertebrates, ganglioside carbohydrate composition determines blood groups. So the ganglioside carbohydrates, it's carbohydrates that are bound to a lipid are what make gangliosides. We'll see that in the lipid chapter. And that's what basically governs what blood group you have, is what different sugars you have attached to this ganglioside. So in gram-negative bacteria, lipopolysaccharides cover the peptidoglycan layer. And what is that peptidoglycan? Huh? Yeah, so it's endotoxin, right, or exotoxin, or LPS, lipopolysaccharides. Yeah. And this is a bacterial lipopolysaccharide, so you have lipids and sugars with a bunch of other sugars attached to it, basically. So you have your lipid A layer, a core, and then your O-specific chain, right? Glycoconjugates are proteoglycans. Sulfated glucosaminoglycans attached to a large rod-shaped protein in the cell membrane. Cytokines, protein has a single transmembrane domain. Glypicans, or protein, is anchored to a lipid membrane, interacts with a variety of receptors, neighboring cells, and regulates cell growth. And this is examples of those. So you have the membrane, the C-terminal side of the protein, cleavage site, which can interact on the outside. Chondroitin sulfate forms here, and then you have your core protein and pepperin sulfate form that extracellular matrix. Or you could have a GPI anchor, which is attached to a lipid embedded into the proteoglycans, such as glypican in the extracellular domain there. And this shows you the different domains where you get glycosylation on a protein. So proteoglycans, different glycosaminoglycans are linked to the core protein. Linkage from anomeric carbon of xylose to serine is the hydroxyl group on serine. Our tissues have many different core proteins. Aggrecan is the best studied. And you have your carboxy terminus and amino terminus, and it's all linked chondroitin sulfate to xylose to the serine near the amino terminus of the core protein. So hyaluron and aggrecan can form these huge complexes of non-covalent aggregates. They'll hold a lot of water, which provides lubrication, very low friction material, covers the joint surface for articulate cartilage, friction and load balancing. This is what I was talking about You can squeeze that water out of it if it's placed under force, and then when you release the force, that water will go back in. So it's a way to add shock. And this is what the structure looks like. Hyaluron, up to 50,000 repeating disaccharide units. Next to the aggrecan core protein, and they kind of look like fans from a fan, or like almost like feathers, huh? So the extracellular matrix is the material outside the cell, has strength, elasticity, and physical barrier in the tissues. The main components are proteoglycan aggregates, collagen fibers, and elastin. The extracellular matrix, a barrier for tumor cells seeking to invade new tissues, and some tumor cells secrete heparinase, which will help degrade that extracellular matrix so they can invade different tissues. So some integral protein, membrane proteins are proteoglycans, it's syndicans. Other integral proteins are receptors for extracellular proteoglycans or integrins. Proteins link cellular cytoskeleton to the extracellular matrix and transmit signals for the cell to regulate. So you can regulate cell growth, cell mobility, and apoptosis and wound healing. So it's a way to link cell signaling to the extracellular matrix. And that just shows you the overall structure of integrins interacting with the extracellular matrix. And you can get a lot of stores of growth factors in the extracellular matrix to help signal those cells. So oligosaccharides and recognition. So this is the role of oligosaccharides in the recognition events outside the cell. So you have an oligosaccharide chain. A lot of viruses have evolved to have their protein coat recognize certain oligosaccharides and allow it to come into the cell that way. And same thing with leukocytes can interact with certain proteoglycans, P-selectin, and then toxins will also. So bacterial toxins can interact with proteoglycans. Viruses can infect the animals. Some bacteria, H. pylori, adhere to the cell and colonize or infect animal cells via proteoglycans, which is what D is supposed to represent. And then E represents selectins or lectins in the plasma membrane, and certain cells mediate cell-to-cell interactions. So you can analyze glycoconjugates. So if you're working in industry, a lot of times you're producing recombinant proteins. If you're producing them in bacteria, you're not going to get any of these, right? Because they don't glycosylate their proteins, which can hamper functions of a lot of recombinant proteins if you're using them for purposes. Some of them need these, sugars added to the proteins. So insect cells or mammalian cell culture will add the glucoses to those recombinant proteins. But you have to analyze how they're added to make sure they match the kind of glycosylation you would need to see the right function for that recombinant protein. So, and in this example, this is mass spec, and they're looking at, this is M to Z is your mass to charge ratio, because mass spec works by, charge repelling down, and it's mass, and separates things by their mass and charge, and you can analyze a specific protein for its glycosylation sites and what glycosylations it has.